Now let's come towards question number 13, that is differentiate between. Basically in this question we have 4 parts, A, B, C and D. We have to differentiate between the given terms. So the first question or the first part in the question number 13 we have is to differentiate between hypocotyle and epicotyle. So what is the difference between hypocotyle and epicotyle? So it is the portion. If you are talking about hypocotyle, it is the portion present on the embryonic axis or embryonal axis just below the cotyledon in a dicot seed. It is the portion present on the embryonal axis just below the cotyledon in a dicot seed. And if we talk about the epicotyle, then epicotyle is also a portion present on the embryonal axis of dicotyledon seed, but it is present just above the cotyledons. Alright, so this is the basic difference between hypocotyle and epicotyle. Also, hypocotyle terminates into radicle and the epicotyle terminates into plumule. So if we write the difference between these two, in the left column we will be talking about hypocotyle and in the right column we will be talking about epicotyle. The basic diagram of cotyledon and embryonal axis is just this kind of thing. These are the two cotyledons showing dicotyledon seeds. Now this is the embryonal axis, the portion below the cotyledons, in a dicotyledon seed on the embryonal axis represents hypocotyle. So this is the portion present on the embryonal axis just below the cotyledons, in a dicot seed is actually known as hypocotyle. And if you talk about the epicotyle, then this is the portion present on the embryonal axis lies just above the cotyledons. In a dicot seed. Also, the closing difference or the second difference between hypocotyle and epicotyle is that this hypocotyle region actually terminates into the region that is known as radicle, which will give rise to the root system of a plant. Alright, so this hypocotyle terminates into radicle. This terminates into radicle, which will give rise to root system of a plant, and this epicotyle terminates into the region known as plumule, and this plumule is going to give rise to shoot system of the plant. As you can see on the embryonal axis of a dicotyledon seed, you can see this is the region just above the cotyledon is known as epicotyle, whereas the region just below the cotyledon on the embryonal axis is known as hypocotyle. This hypocotyle is terminating into radicle, and this epicotyle is terminating into plumule, which will be giving rise to shoot system of a plant, and this radicle is giving rise to the root system of a plant. So this is the basic difference between hypocotyle and epicotyle. Now let's move on the second part of this question, that is differentiate between coleoptile and coleorrhiza. So what is the difference between coleoptile and coleorrhiza? This, if we talk about coleoptile, it is just the conical protective sheath. It is a conical protective sheath in case of monocot or monocotyledon seed, which is enclosing the plumule part. So this coleoptile, in the left column we are going to discuss about coleoptile, and in the right column we are going to discuss coleorrhiza. This coleorrhiza and coleoptile are the parts of monocot seeds, not dicot seeds. Alright. So what is the difference between coleoptile and coleorrhiza? It is a conical protective sheath enclosing the plumule part of a monocot seed. Alright, and if we talk about the coleorrhiza, it is also a protective layer enclosing the root cap and radicle in the monocotyledon seed. Alright, so this is the basic difference between coleoptile and coleorrhiza, and these are the parts of monocot seeds only. So coleoptile is a conical protective sheath enclosing the plumule, of a monocot seed. Alright, coming on to the coleorrhiza. What is coleorrhiza? It is also a protective covering which is enclosing radicle as well as root cap in the monocot seed. So it is also a protective layer enclosing the radicle and root cap of a monocot seed. Alright, so it is also a protective layer which is covering or enclosing the radicle and root cap of a monocot seed. Why monocot seed? This is only present in monocot seed only. This is not present in dicot seed. Coleorrhiza and coleoptile are the parts of monocot seed. They are not present as protective layers in dicot seed. So this is the basic difference between coleoptile and coleorrhiza. Now moving on to the third part of question number 13, that is differentiate between integuments and testa. Now what is the difference between integuments? You must have heard about integuments. Alright, what is integument? We know that we find integuments as the covering, the outermost covering of an ovule. What is ovule? The female megasporangium present inside the ovarian cavity or a locule. Alright, so this ovule is present inside the locule or an ovarian cavity. This ovule has hard coverings. This ovule has hard protective coverings, and these protective coverings of ovule is known as integuments. Alright. Now if we talk about the testa, testa is the outermost protective covering of a seed. Testa is just the outermost protective covering of a seed. Both are the coverings. Integuments is also a covering, testa is also a covering, but integument is a covering of ovule, that is female megasporangium, and testa is the covering of a seed after fertilization. Although this testa has been made from the integuments only. After fertilization, what happens in a flower or angiospermic plants? After fertilization, what happens to the ovule? That ovule along with its integuments convert into seed and its seed cover. The ovule, that is megasporangium of a female, converts into seeds. Ovule gets converted into seed after fertilization, and the ovary converts into fruit after fertilization. What happens with the integuments of an ovule? That integuments of an ovule converts into the protective layers, protective coverings of seed, that is testa. These integuments are going to convert into testa only. Alright, so the difference between testa and integument is, first we are going to talk about integuments. Now what are integuments? These are the outermost protective layers or coverings of a female megasporangium, that is ovule. We know that if we draw a diagram of an ovule, it looks like this. This having coverings. What are these coverings? These are integuments only. These integuments after fertilization, along with the embryo sac, along with the ovule, which is converting into seed, these integuments are converting into the seed coat, which is known as testa. Now let's talk about the testa. What is testa? First of all, it is the seed coat or outermost covering of a seed. This is the outermost covering of a seed which is developed from integuments only, After fertilization, the integuments convert into testa, which is the protective covering, hard covering seed coat. Alright, the covering of seed coat. So this is the basic difference between integuments and testa. Now moving on to the last part of this question, that is differentiate between perisperm and pericarp. Now we will be talking about the perisperm and pericarp. First we are going to discuss perisperm. What is perisperm? So the persistent residual nucellus. Now what is nucellus? We know that nucellus is the special kind of tissue which is present inside the ovule. Alright, so what happens after fertilization? This nucellus tissue is actually digested by the developing embryo. In some seeds it does not. It persists for time. Alright, so what is perisperm? The residual or the persistent residual nucellus in some seeds. That persistent nucellus is actually known as perisperm. It present in some seeds like beet or black pepper. Alright, so this persistent nucellus tissue in some seeds is known as perisperm. Whereas if we talk about what is pericarp, it is the ripened wall of a fruit. Pericarp is just the ripened wall, outer covering of a fruit which is developed from the covering of ovary. Alright, which is developed from the covering of ovary. Let's talk about perisperm. Perisperm here we are going to talk about. Then here In the column right, we are going to talk about pericarp. Don't get confused with perisperm and pericum, as both terms include peri. Alright, so this perisperm, if we talk about perisperm, then this is the persistent residual nucellus tissue. Alright, this is the definition of perisperm. What is perisperm? This is the persistent nucellus tissue in the seed, which is known as perisperm. It is present in we are giving example of this. It is present in some seeds like beet and black pepper. Alright, black pepper. These are the examples inside which the seeds of black pepper and beet have residual nucellus, persistent nucellus in them. That is why we call them as perisperm, because after fertilization all things get changed, all things get converted into something. So similarly here, the nucellus tissue of an ovule is converting into perisperm, which is present in some of the seeds such as black seed and black pepper and beet. So this is perisperm. And if we talk about pericarp, this is the ripened wall of a fruit. This is a ripened wall of a fruit. It is developed from the wall of ovary. Because we know that after fertilization, ovary gets converted into fruit, ovule gets converted into seed. As we know that the outermost covering of an ovule, which is known as integument, converts into testa, that is the covering of seed after fertilization. Similarly, in case of ovary, we know that ovary has a wall. After fertilization, when ovary converts into fruit, the wall of ovary also converted into the wall of fruit, that is known as pericarp. So this is all about the question number 13, and we have completed all the parts A, B, C and D, all the parts of question number 13. With this, we have completed the solution of question number 13.